Hello, class. This is Professor Fisher, and this is Chapter 7, DNA Structure and Gene Function. And so we want to study how, what DNA is, deoxyribonucleic acid, uh, and why it's important. DNA is important because it transfers genetic information from one generation to the next, but it also codes for who we are. It's sort of like the roadmap uh, or the code for our life. So what is DNA? DNA is it's a molecule, right? And so we've talked about lipids, we've talked about carbohydrates, we've talked about proteins, we've also talked about nucleic acids. DNA is a nucleic acid. It's made up of nucleotides, but carbohydrates, we tend to think of them as energy. We break the bonds for different carbohydrates, we make energy, versus DNA, as you see here, is more so related to having a code. All right, we want to know the code of the DNA. How did scientists discover DNA? Well, it was, as you see here, is one of the experiments. It was a lot of work done to be able to discover what DNA was, but using bacteria, you could see that good bacteria versus bad bacteria, um, that DNA was, they figured out it was DNA that transmitted the information between toxic and harmless bacteria. So using bacteria, they were able to find DNA or figure out DNA. Now, DNA eventually codes for protein. So how do we know it was the DNA and not the protein? Well, using viruses, we were able to figure out that it was DNA and not protein that contained the genetic information. And we know now that DNA codes for the amino acids that are in protein. DNA is composed of nucleotides, right? So nucleotides. One phosphate group, as you see here, one molecule of 5-carbon sugar, a deoxyribose, that's where the name, the D stands for, and then one nitrogenous base, adenine, guanine, cytosine, or thymine, right? That's the structure of DNA. So a phosphate group, a sugar, and a nitrogenous base. Nucleotides join together into strands. So we have these long strands, a sequence of nucleotides, right? These long strands uh, that form to make up what is called DNA. Lots of nucleotides, lots of information. DNA is a double helix, right? So that's just talking about the shape of the DNA. So one DNA molecule is made up of 2 strands of nucleotides. So you see the nucleotides on each side, and they twist as they bind to each other. And so you have this double. They call it a double helix. DNA strands are held together by base pairing. Right, so we have these hydrogen bonds, which we've mentioned hydrogen bonds before when we referred to water molecules. We mentioned hydrogen bonds, but we see them here and also DNA strands. And so adenine, the nitrogenous base adenine binds with thymine, and guanine binds with cytosine, and that's how DNA binds together. All right, the nitrogenous bases connect together with hydrogen bonds. Right, very complex molecule. The hydrogen bonds can only form between complementary base pairs. A binds with T, G binds with C, right? So the nucleotide sequence in one strand determines the sequence in the other strand because they can only bind. You see a double bond and a triple bond. So they bind with complementary base pairs. And so DNA strands are anti-parallel. Each strand of DNA is, each strand of DNA, each end of the DNA strand is different. Uh, and they're anti-parallel. So the strands are anti-parallel, oriented in different directions, right? So kind of like one is upside and one is upside down, and they're bound together, anti-parallel to each other. So One is going one way, one is going the other way, so to speak. It's like two roads. Uh, one is going on two sides of a road. One side is going one direction, one side is going to the other direction, but they're running parallel to each other. So that's called anti-parallel. Right? Protein is what we ultimately get from DNA. Is protein, right? So, like I said, things like glucose, carbohydrates give us energy. And ultimately, from a molecule of glucose, we get ATP, 36 molecules of ATP, adenosine triphosphate, which can be used as energy for the cell. For DNA, the ultimate goal, or what we get from DNA, is protein. Now, we don't break it down like we do with glucose, but we can take the code, which we get from the nucleotides, the code, uh, the combinations of the A, the T, the G, and the C, the code codes for protein. So a gene is a small region of a chromosome, and this is where it can get a little confusing, but think about it. So a gene is a small region of a chromosome. The sequence of DNA in each gene encodes a specific protein. So a gene is a section of DNA, which is your total DNA is all of your chromosomes, but a section of your DNA makes up a gene, and that section codes for a protein, right? RNA is another term that we're going to see. RNA, uh, ribonucleic acid, RNA, is very similar to DNA, but instead of deoxyribonucleic acid, we have just ribonucleic acid, all right? So RNA and DNA are very similar. The sugar in DNA is deoxyribose. The sugar in RNA is ribose. RNA has a different one of the different nucleotide bases too. So not only does it have a different sugar, but it also has a different base, uracil, and a slightly different form. DNA is double-stranded helix. RNA is single-stranded. The purpose of RNA is that DNA stores our genetic information, but when we're going to copy a section of DNA, copy a gene, so that we can use that copy to make protein, we don't copy both strands of the DNA. We copy one strand of the DNA into a single strand of RNA. So just as long as we need. So remember, a section of DNA that codes for a specific protein is called a gene. So we copy that gene, just one side of the DNA, into RNA, and we can use that to make, now we have that code for just that one gene, and we can use that to make the protein. This slide here is kind of cool. Making proteins is like making brownies. So protein production occurs in two stages. We have what is called transcription, and then we also have what is called translation, right? So transcription is when we copy a section of DNA called a gene into RNA. Translation is when we take that code that is now in the RNA and we use that to translate it into a polypeptide chain, into the amino acids that make up a protein. Right, so the first step is transcription, is RNA synthesis. Transcription uses DNA, right? So the DNA stays in the nucleus. We don't want the DNA leaving the nucleus. So transcription uses DNA as a template. The nucleotide sequence of the DNA determines the nucleotide sequence of the RNA. And so the RNA sequence is complementary to the DNA. So we're copying the DNA. Translation takes place at the ribosome. So once we've copied a section of the DNA, a gene, it exits the nucleus, and at the site of a ribosome, it can then, with the help of messenger RNA, with the help of ribosomal RNA and transfer RNA, it can then carry out translation, which is converting that code into protein. So converting that code into protein, getting the necessary amino acids that RNA code tells us, and what protein is a chain of amino acids. The code for the amino acid chain comes from the RNA, which was copied from the DNA. So let's look at how this happens. So transcription occurs in the nucleus. To make RNA, we have a little bit different nucleotide base pairs. So adenine pairs with uracil, cytosine pairs with guanine, guanine pairs with cytosine, and thymine with adenine. Right. So the code for the DNA will pair with these base pairs in the RNA. Transcription occurs in 3 steps: initiation, elongation, and termination. And this is transcription is copying the DNA. So we have this RNA polymerase, this enzyme that gets everything started. It binds to the promoter region of the DNA, which is the beginning of the gene, and unwinds the 2 strands of DNA, because we only need to copy one, because remember, they're complementary to each other. That's the DNA. DNA is the template, so we only need to copy one. RNA polymerase moves along that strand of DNA, copying one of the DNA, and makes an RNA copy. Elongation, the RNA nucleotide base pair with the template strand. RNA polymerase joins them together. So we have a copy of one of the strands of the DNA. RNA is anti-parallel to the DNA. So remember, we're matching it with its opposite, with what it binds to, right? So it's anti-parallel, just like the DNA is anti-parallel. So the 3' end of the RNA matches the 5' end of the DNA. So we're matching it with the one it binds to. The RNA polymerase reaches the terminator, the end, which is found at the end of the gene. So we've copied the section of DNA that corresponds to the gene. At the terminator, the RNA, DNA, and RNA polymerase separate from each other, and the DNA becomes a double helix again. The DNA goes back to being normal DNA, but now we have a section called RNA. It's not ready yet. The cell must modify the RNA before it can carry out its function. A cap structure is added to the 5' end, a poly A tail structure is added to the 3' end, so it's just doing some processing to make it just right. Introns, there's intron sequences in the genes that are not used for producing a protein, and there are exons, which are sequences that specify amino acids. So some of the copy of RNA we don't need. Your body knows which part that is. Those are called introns. The part that we do need are called exons. So we need to remove the intron section of the RNA, the sections that we don't need. Now the processed RNA is a functional molecule, and when processing is complete, it can leave the DNA, leave the nucleus. Your DNA stays in the nucleus, but this RNA copy of a section of DNA called a gene can now leave the nucleus. Now let's look at how it becomes a protein. Translation. So transcription, we went from DNA to RNA. We actually call the RNA that we go to mRNA, messenger RNA. It takes the message from the DNA, so we call it messenger RNA. But then we also have in at the site of the ribosome, we have what is called translation, right? So we take that, we take that, we're going to use that genetic code from that was originally from the DNA and code it into, we want to, every 3 nucleotides, so AGUC, every 3 nucleotides codes for a specific amino acid, or we call it, you see here, polypeptide, which is going to be an amino acid sequence. So a sequence of amino acids. So each codon, each 3 nucleotides codes for a specific amino acid. Those amino acids, when they're bound together, give us the protein. So mRNA codes for proteins, and it tells you which proteins it codes for right here. tRNA translates the genetic code. So we have the RNA that we copy from the DNA, we call that mRNA, messenger RNA. We also have tRNA, transfer RNA molecules. Transfers RNA molecules bring the amino acids to the ribosome. So it actually brings each amino acid that the mRNA says, that the mRNA is telling us is coded for with that codon. So tRNAs are adapters that recognize the genetic code and bring the appropriate amino acid so that we can put it in place. tRNA matches up to the mRNA, so it binds, tRNA binds to the mRNA codon and the corresponding amino acid. So each step in translation happens at the ribosome. Ribosomes, remember, those are membrane, and those are the site of protein synthesis in the cell. And so ribosomes help the 3 types of RNA interact with each other to build a protein. The large subunit binds to tRNA, the small subunit binds to mRNA, and we're able to build proteins. So translation occurs in 3 steps: initiation, elongation, and termination. Very similar to transcription, right? So translation, we start off with small ribosomal subunit binds to mRNA. Large ribosomal subunit binds, and an initiator tRNA molecule binds the anticodon with codon. So we have anticodon, see it here, amino acid, it brings the amino acid. The large ribosomal subunit here, we have our RNA here, right, which has our sequence, our code. The initiator tRNA anticodon complementary base pairs to the mRNA start the codon. That initiator tRNA already carries the amino acid MET, M-E-T, you see there, amino acid. This is the first one. The second tRNA enters the ribosome next to the initiator, the second one here. Its anticodon matches the second mRNA codon. So it matches, this is the mRNA, this is the tRNA here, matches this code and brings the next amino acid, and they join together by a peptide bond, starting to build something here. The first tRNA leaves the ribosome, but its amino acid stays behind. The ribosome moves to the right. The ribosome is this big unit here. It's moving down this RNA, and messenger RNA, and slowly each 3 nucleotides going to bind to a section of tRNA, which is going to it's going to bind to the next next amino acid. Once it reaches the end, it reaches a stop codon, the last three, the stop codon at the end of the gene, and this causes the protein to release. A protein called release factor binds to the stop codon. No tRNA for that, and it's going to stop building the amino acid chain. The polypeptide detaches from the mRNA and folds into a functional protein. And that's how we get to protein. Transcription to translation to protein. DNA to RNA to protein. Translation is very efficient. Multiple ribosomes can attach to an mRNA molecule. So you can have multiple ribosomes simultaneously making many molecules of protein all at once. So we can be continuously building proteins from one mRNA molecule. Protein synthesis is highly regulated by the body. Protein synthesis requires a lot of energy. Remember, this is an anabolic reaction. You're building, so it requires energy to build protein. So cells save energy by only producing needed proteins. But we need that. This is one of the things your body has to do at all times, building. But this has to be regulated by the body. Prokaryotes regulate several genes at once. So prokaryotes, remember, they have their DNA floating around. Genes in prokaryotes are organized into what's called operons. Groups of genes are always transcribed together. Operons contain related genes, so related genes will be transcribed together. So I need the gene for the nose hairs, and smell, for instance. That's a bad example, but they would all be transcribed together. Promoter, operator, control gene expression. The promoter is the region of DNA where RNA polymerase binds to initiate transcription. The operator is the region of DNA where there's stuff with the regulatory binds, and which can affect the activity of the RNA polymerase. Then you also have repressor proteins, which can block transcription. Right? So we want to be able to control transcription, control when a molecule of DNA is transcribed. But we have proteins that can block RNA polymerase from binding to the DNA, therefore blocking transcription of DNA into RNA, right? So if we don't need a specific protein, or if a prokaryote doesn't need a specific protein, this is how they would block it. Let's talk more about that. Eukaryotes have multiple levels of gene regulation. Eukaryotes, that's us, human beings. It starts in the nucleus. Some genes are wound up very tightly, cannot be used for transcription, but other genes are available. Remember, you have a lot of DNA in your body, so you have multiple, many genes on these many strands of DNA in your body. So you have many different proteins called transcription factors can bind to a gene, bind to a specific location on the DNA to affect the activity of RNA polymerase. Remember, you need RNA polymerase to be able to transcribe that DNA. Transcription of the gene can only occur if the correct transcription factors are present. So these transcription factors help us have the ability to be able to transcribe, to take that DNA and to copy it into mRNA, messenger RNA. The RNA polymerase, the enzyme RNA polymerase, needs these transcription factors. So this is how your body how eukaryotes, but also human beings, because we are eukaryotes, we are one of the eukaryotes, how we can control transcription. So these transcription factors, RNA polymerase needs these transcription factors to bind to the DNA, to the gene, in order for that gene to be transcribed. So eukaryotic genes can be regulated by splicing as well. So some eukaryotic genes can encode multiple proteins by using different combinations of exons. Alternative splicing creates different proteins from the same mRNA, so it's very efficient. So if I need a certain protein, I'll cut out, I can use one copy of mRNA and use that to make different proteins depending on how I process that mRNA. RNA export can also be regulated. So multiple ways, transcription factors, how we process the RNA, the mRNA, but also how we export the mRNA. So gene regulation continues in the cytoplasm, but certain eukaryotic proteins can hold mRNA inside the nucleus, preventing them from reaching a ribosome. If the mRNA doesn't reach a ribosome, then we don't have translation. So we can control it. That's another way that this process can be controlled. Some mRNA may be quickly degraded before it can be translated into protein. Another way it can be controlled. Proteins must be properly folded before they are functional. They must also reach their correct cellular location. So if a protein is not properly folded or modified in and of itself, we can have regulation of that protein that way. For protein, we talked about the body having a specific pH and temperature and all those things, because if it doesn't, it would cause things to denature in the body. So if something we don't need in the body, that thing can be modified. Your body can use chemicals to denature it, change the pH on it, denature it, and your body will no longer be able to use it. So if I have too much of a certain protein in the cell, I can denature those proteins and they won't do anything. DNA can also mutate. Unfortunately, this can cause many problems, but it is something that is important for us to study. A mutation is a change in a cell's DNA sequence. Now, we saw that DNA, each codon set of 3 nucleotides will code for a specific amino acid. So all I have to do is change one nucleotide, and I could change the amino acid, which would then change the protein. And so you can see how easily per se it would be to mutate DNA, because a different amino acid chain makes a different protein which has a different function. Here on the page you see a mutation can come in several varieties. Here we see legs where there was an antenna. We have a point mutation, changes one or a few base pairs in a gene, which is what I was just talking about, one or a few base pairs changing. Nucleotide substitutions cause small changes in the protein structure, right? So changing a single nucleotide, which would change a single codon, which would change a single amino acid. But some mutations can cause disease. So a single base substitution in a hemoglobin gene can cause blood cells to form abnormally, leading to what is called sickle cell anemia. So some mutations can actually cause disease. You also have frame shift mutations, which cause large changes in protein structure. So insertion of one nucleotide. So if instead of changing a single nucleotide from like a A to a G, I added a nucleotide. So then everything after that nucleotide would be off. If I added an extra nucleotide, that would be frame shift. So that would affect multiple amino acids, as you see here. Somebody, if I have, if the protein is normally 10 amino acids long, and I add a nucleotide, now I could have if I added it in the middle, I could have 5 different amino acids. So it would change half my protein. That would be a major mutation. Mutations can create genetic variability. Sometimes they're not always harmful. Sometimes they give us a few differences here and there. And here we have just investigating life. We talk about how just by a few amino acids difference, we have much different genes, which are the difference between a human, a chimpanzee, a gorilla, or an orangutan. And so some people think there were just mutations between um, some of these other animals and human beings, or some people think um, the same being created these different beings and created them somewhat similar. I'll leave that to I'll leave that for you to think about and to ponder over. But this is DNA, and this is transcription and translation and genes, nucleotides, and all the things. Hope you have enjoyed this lecture.